Okay, let's dive right in and unpack the chemical magic of hematoxylin. Welcome to today's explainer. We're going to demystify the absolute most common biological stain in the entire world. It's kind of crazy to think about, but every single working day, literally millions of microscope slides are prepped globally using a technique that's been around for over a century. And today, we're going to figure out exactly how it actually works. Now, you know those vibrant purple and pink colors you always see on pathology samples? Well, they aren't natural. They're actually the result of this incredibly brilliant chemical trick designed to reveal totally invisible cells. Take a standard 5-micron section of a renal cell carcinoma, for example, stained with hematoxylin and eosin, good old H&E. The pinks you're used to seeing? That's the acidic eosin grabbing onto the cytoplasm and connective tissue. But those sharp, dark, blue-purple dots? Yep, those are the cell nuclei, and they are exclusively colored by hematoxylin. It is widely, and I mean absolutely widely, considered the ultimate special stain for the nucleus. Which naturally brings us to a really fascinating question, one that's going to guide our whole molecular zoom-in today. How exactly does this dye seek out and attach itself to the nucleus of a cell? I mean, the inside of a cell is a highly complex, crowded environment. So how does this one specific chemical know exactly where to go and then hold on so tightly that we can see it clear as day under a microscope? Section 1, From Tree to Lab. It's pretty rare in modern, high-tech lab settings to rely on something completely organic. But that is exactly where this story begins. Long before it ever saw the inside of a diagnostics lab, this dye was actually extracted straight from a Caribbean tree. Specifically, a tree with this incredibly hard heartwood that ranges anywhere from dark orange to a deep purplish red. We're talking about the Hematoxylin campechianum, or as it's more commonly known, the logwood tree. Historically, it was used by native populations in the Caribbean way before making its grand entrance into histology labs back in the 1830s. Now, the crude logwood extract itself is a mix of things: tannins, resins, volatile oils. But it's that pure hematoxylin compound we are really after to get that perfect cellular stain. Section 2. The chemical matchmaker. Here's the kicker. Straight out of the tree, hematoxylin actually cannot stain tissue at all. It needs a little bit of help. The transformation it has to go through is just so elegant. First, the dye has to be oxidized into hematane. Then it's combined with a metal to completely flip its electrical charge. See, here's the fundamental problem. Right after oxidation, hematine is anionic. It carries a negative charge. And the DNA packed inside a cell nucleus, well, it's absolutely full of phosphate groups, which are also negatively charged. Basic physics, right? If you try to force a negatively charged dye onto negatively charged DNA, they're just going to repel each other. No binding whatsoever. So we desperately need a bridge. Enter the mordant. This is our crucial chemical matchmaker that completely solves the repelling issue. In your routine H&E staining, this mordant is an aluminum, specifically aluminum double sulfate. When you introduce this aluminum mordant to the hematane, something really dramatic happens. The metal atom physically incorporates right into the dye, creating this positively charged cationic complex that we call a lake. Suddenly, just like that, our dye is primed, supercharged, and ready to hunt down those negative charges in the tissue. Section 3. Binding to the nucleus. Now we're zooming all the way in, because this is where the true chemical magic happens. Inside that microscopic landscape. Once we have our positively charged lake, it literally acts like a molecular magnet. It makes a beeline straight for the phosphate groups in the DNA strands repeating sequence. Because remember, DNA and RNA are highly acidic thanks to those phosphoryl groups. So that positively charged aluminum dye complex is deeply attracted to these negative targets. But, and this is key, it doesn't just stick there statically through electrical attraction alone. It actually forms highly specific chemical bonds that lock it tightly into place. The actual attachment of this mordant metal to the tissue happens through a process called chelation. Think of the aluminum metal as a highly specific lock and key. It forms incredibly strong covalent and coordinate bonds with the tissue's phosphate hydroxyl groups. Specifically, a covalent bond forms with a hydroxyl oxygen, and a coordinate bond hooks up with an electron-donating oxygen atom. The aluminum is basically acting as the ultimate anchor, bridging the dye and the DNA, holding the whole shebang together with unbelievable stability. But wait, it's not just electrical attraction and chemical bonding doing all the heavy lifting here. The second law of thermodynamics actually jumps into the mix too. It plays a massive role in maximizing disorder, which is what actively drives the dye so deeply into the cellular structures. As the dye interacts with the tissue, hydrophobic bonding really helps the dye's affinity in aqueous solutions. Because the system naturally wants to maximize its own disorder, it effectively pushes and shoves the dye molecules to disperse thoroughly across the entire tissue matrix. That's how you guarantee such an even, incredibly deep stain of all those nuclear components. Section 4, the perfect recipe. Now, obviously, not every single tissue sample needs the exact same approach, right? So over the decades, pathologists have tweaked this chemistry to fit totally different lab needs. If we look at 3 of the most popular formulations, the data paints a really clear picture. Hematoxylin, for instance, has a very high mordant content, which gives it a much firmer bond and, frankly, superior nuclear staining. It uses chemical oxidation, usually something like mercuric oxide or potassium iodate, so boom, it's ready to use right away. Because there's way more mordant than dye, it holds on tight. On the flip side, you've got Mayer's and Ehrlich's solutions. These have higher dye-to-mordant ratios, which actually creates a much looser bond. Ehrlich's is especially wild because it can be ripened completely naturally. You literally just leave it exposed to sunlight and air for 2 to 3 months. Talk about old school. And this specific ratio of dye to mordant totally dictates how a tech actually stains the slide in the lab. You basically have 2 choices. You either gently build up the color progressively, or you go all in, regressively overstain the slide, and then carefully peel the color back using acid. Solutions like Mayer's are progressive. They're milder, selectively staining just the nuclear material a beautiful deep blue without making a mess of the rest of the slide. But solutions like Harris and Ehrlich's, they're regressive. They aggressively overstain everything. So to get that crisp, clear cellular picture, a technician has to wash the slide in a dilute acid. That acid breaks the weaker bonds in the cytoplasm, but leaves those firmly bound nuclear lakes totally intact, shifting the background color beautifully from a murky purple to a clean salmon pink. So I'll leave you with this final thought to chew on. Despite every single modern advancement we've made in immunohistochemistry and molecular biology, this elegant 19th century chemical reaction is still the undisputed champion of the histology lab. I mean, even when researchers are using the newest, most cutting-edge antigen retrieval protocols, they almost exclusively still rely on good old alum hematoxylin to counterstain the nucleus. It's just a huge testament to the absolute perfection of this natural dye and its brilliant chemical matchmaking. So next time you happen to see a medical slide, take a quick second to appreciate the centuries of natural history and thermodynamics working in perfect harmony inside those tiny, dark purple dots.